logistics after. Okay, great.
All right.
Okay, welcome back. So we are going to continue with the short talks. It's 15 minutes. So first speaker is Noemi Picco from Swansea University. Noemi.
Thank you. Thank you very much. Can you hear me?
Yes.
Yeah, good. Okay, yeah, so I'm interested in environmentally mediated drug resistance, and particularly in the context of molecular inhibitors. So I'm going to look specifically at those molecularly targeted inhibitors that go after the driver mutation in a cancer, such as BRAF inhibitor, EGFR inhibitor. These have been proven to be quite successful. At least initially, they induce quite a good response, especially compared to chemotherapeutic. Unfortunately, these response is just temporary. And so this is kind of a typical clinical scenario that we would observe. We would see initial good response, which is then followed by a slow progression. And if we interrupt the treatment at a disease progression, then we would observe some explosive growth that can cause the so-called disease flare. If we interpret this in the classical way, so ecologists would interpret it in the most simplistic way, which is the therapy has been selectively killing the sensitive portion of the tumor, leaving behind the resistant part exposed to more resources, which of course then would grow slowly but steadily. If we were to reintroduce the treatment, then we will still observe some response. So this hints at some version of mechanism of reversibility that we can perhaps exploit. So these, as I mentioned, there's the simplistic explanation is the sensitive versus resistance. But if we really want to go to dig deeper into the causes for this kind of pattern of response, then we cannot just look at the tumor in isolation. We need to look into the tumor microenvironment, because this is where various components of the tumor microenvironment will contribute and be the key players in a number of compensatory mechanisms that come into place once we give this therapy. So some of these so-called adaptive responses, they vary from phenotypic switching, secretion of some factors such as growth factors, signaling molecules that would reactivate alternative proliferating pathways, metabolic reprogramming, and of course the immune response. All of these are being studied by several people. For the here and now, we'll focus really on the top two. And the good news about these kind of responses that I'm going to look in, as we heard earlier as well, is that they're just temporary and they can be reversed. They can be reversible. So this is sort of the idea behind introducing the rationale behind introducing some pauses in the treatment, so we allow enough time for the tumor microenvironment to renormalize. So intermittent treatment has been successfully employed in preclinical studies. As far as clinical studies go, there have been some mixed outcomes, with patients responding really well, like this one, and other patients just really not doing any better than continuous treatment. And then a phase 2 trial definitely ruled out intermittent treatment because it wasn't really no better overall than the continuous treatment. But still, we see really good response in some patients and really bad in others. So the question here now is to look maybe at the underlying mechanism, so we can try and explain this variability across patients. But also this tells us that we don't really yet understand much of the mechanisms behind. And that's where the modeling can come in handy. So I'll start, because this process includes a number of complexities in terms of spatial, temporal scales, key players, processes at play. I'm going to start with a very simple model, which is making use of a very simple experimental model as well, and then gradually build up complexity. So the initial very simple model is an OD model, which just mimics the change of state between cancer cells sensitive to resistance and then stroma cell from active to reactive and inactivated again. And in particular, this was looking at cancer-associated fibroblasts. Now, the experimental setup is a couple of BRAF mutated cell lines, melanoma cell lines, which are being treated either on a dish or in the mouse. And the idea is that with this simple model, we can capture the two portions of the growth dynamics and basically mirror them to each individual experimental setup. So specifically, the in vitro experiments, the dish experiments, they capture very well the exponential initial phase of the growth. So we will use those to calibrate the exponential phase. And these are run in a couple of days worth of time. But if we zoom out in the time scale and we look over longer time scales, then of course the logistic component comes into play. And this is better captured by the in vivo experiment, where obviously the tumor is growing within the constraint of pre-existing tissue. And that's where we could capture and calibrate the logistic-based parameters. And here, the key finding here was that there was a very large heterogeneity in mice that were genetically engineered to be identical. And in particular, this heterogeneity resided in two model parameters, the carrying capacity and the intensity of the tumor stroma crosstalk. And this was the reason why we decided to then go into more details into modeling and understanding these tumor stroma crosstalk, bringing in a new and improved model, which is an individual-based model that you've heard of from many others before. So the setup is quite similar to what everybody else has. The advantage of introducing this is that we can relax a couple of assumptions. So first of all, we're not averaging over space anymore. We can take into account local interactions. Secondly, introducing some stochasticity allows us to capture the emerging dynamics at the tissue level, starting from stochastic cell-to-cell interaction, and therefore the variability and the heterogeneity into the response. But most importantly, particular to this problem, we can explicitly mimic this adaptive response that I talked about. And we do this in the form of a variable P, which is a proliferation signal, which basically integrates all of the signaling cues that the cancer would receive. So these include some pro-apoptotic signaling coming directly from the drug, and then some pro-proliferative signaling coming either from the tumor to itself, so autocrine signaling, or from the direct contact of tumor with reactive stroma in a kind of paracrine fashion. So all of these contrasting signaling cues that will come in and kind of integrated, and the net effect will be that the cell will read the local concentration of proliferation signal and make a decision based on two thresholds about its cell fate. So proliferate, die, or stay inhibited. So if we use this model and we employ intermittent treatment, obviously I'll skip through a lot of this, but here is the crucial thing. The interesting thing is that with some calibration, as much as possible based from the literature, we can find some intermittent treatment schedule where we have some approximate version of control in terms of tumor burden. So after, as you can see, after some initial transient, we get to a point, it's really quite, it doesn't show very well, right? It's quite white. Anyway, there is some bands here that show where we apply the treatment. There you go. Thank you. But what you can see, a corresponding point into successful consecutive treatment poses, that the tumor keeps coming back roughly in the same place. And so now the next question is about what's so special about those spots there? Why is residual disease occurring especially there? So to answer this question, we look first of all at something like the longitudinal occupancy of cancer. So overall, across the entire simulation and over repeated simulation, where are the kind of geographical regions where tumor keeps coming back? And so we do this with cancer. And then if we do the same with the reactive stroma, which are the cells that engage into the crosstalk, we see that there's definitely co-location between cancer and stroma. Not only that, this shows us that basically the cancer needs kind of a critical mass of cancer from the autocrine signaling and the paracrine signaling. We can go a bit more into detail and look at cell neighboring analysis. So to do that, I'm going to distinguish in this kind of sample simulation two arbitrary chosen regions. One is in the middle, where I'm definitely sure there's going to be eradication, and one somewhere else where there's no eradication. So I'm calling these eradication and survival niches. And I want to do differential analysis between the two. So if I look at the average kind of neighbor composition, I'm going to focus on different cell types every time. So now I'm looking across the simulation for any cancer cell that has ever existed in a given position, what is the neighboring profile? So if I look at neighbors that are other cancer cells, then these are the two distributions, and you'll see that they're sort of slightly shifted to the left for the survival niches. So there's a bit more tumor clustering in what then turns out to be the eradication niche. If we then repeat the analysis, but just now counting the passive stroma cells, which I just realized now I didn't mention. So the stroma can be inactive or inactive form, but we also allowed a third kind of stroma, so non-cancer cells, which is unable to engage in crosstalk with the tumor. So this acts purely as a competitor. And so here we want to basically quantify competition by looking at how many cells stick around the cancer in either cells, in either regions. And what we see is that there is a bit of a longer tail in the survival niche. And so there is a bit more spatial competition. So far, the region that we tagged as survival niche doesn't really have the properties of what you would think induce survival, right? So there's a bit less clustering, which is what would provide autocrine signaling, and there's a bit more competition. But if we now look at the reactive stroma cells, it looks like it is them that really tip all these cancer cells into either quiescent or to proliferative, and it doesn't take many. Just a couple of them are sufficient to actually kind of tip all of these across the line. In itself, these two really don't tell us much yet, because what we've seen is just co-location and presence of reactive stroma cell in correspondence with cancer, but doesn't tell us much. In itself, these tissues were constructed to have homogeneous distribution of stroma to begin with. So there's nothing intrinsically different in these two regions. They all had the same kind of raw material to begin with. So there's something else. And so we're looking into another component of the tumor microenvironment, which is the one that breaks this homogeneity, breaks this symmetry, and could explain. And those are the vessels. So they're not quite visible, but there's vessels seated all over the tissue, and they're based on experimentally measured. So this is a realistic tissue with diffusion of the drug coming from the vessels. They're perpendicular to the plane, so it's quite simplistic representation, but their distance is experimentally based. If you think of the vessel as the source of the drug, then of course it would make sense that the closer you are and the sooner you will get killed. Too far, nothing will happen. But the drug is also the source of activation of the stroma. And so there should be a middle range. And here's where the spatial scales become really important. There will be a middle range where you haven't been killed yet, and you get a chance to be rescued by the reactive stroma, which has been activated. There are multiple vessels, so we have to come up with some measure of density. So if we kind of sum up all of these decaying exponential to figure out what is the sort of combined contribution of all of them, you do see in the corresponding regions, which are exactly the same as the one I was looking at before, you will see that there's some increased vessel density. Not sure if you can see from there, but there is increased vessel density there overall. So now we proceed with the hypothesis that by varying tumor vessel density, we could get some nonlinear response in the terms of treatment outcome, keeping the very same intermittent treatment just by varying the vessel density. So we do kind of a thought experiment next. And what we do is varying vessel density, and this is just seeding more vessels, and they're on a very rigid grid. So the results look a bit artificial, but it's just a proof of concept sort of exercise. So we start with very low vessel density. We employ the intermittent treatment, nothing happens, the tumor explodes. Low vessel density doesn't allow enough drug to be perfused into the system. As we increase the vessel density, we get near really good eradication. We increase a bit more, and what we get to this other end here, which is exactly the parameter space in which the simulations I showed you so far were living, which is the sort of a middle ground where we still have resistance occurring, but it's resistance of a very different kind. So we kind of named these two resistant kinds, and from beyond the audience, if there's any biological or medically more inclined people that will suggest better terms, then they're welcome. But we sort of define these as malperfusion driven resistance, because it's resistant just coming out of the fact that the tissue cannot be reached with the drug they want to give it. And on the other end is the stroma driven resistance. So the tissue is well perfused, the drug makes it there, but there's the EMDR, the stroma that comes in and kind of counteracts. I'm not sure how I'm doing with the time because I can't really tell when I start. Okay. Anyway, so I think I'll just get to the end very quickly. This is the next step here, obviously. Sorry, I'm supposed to go back. So the next step will be to actually introduce a bit more realism. And the first thing that we really need is to consider the heterogeneity within the cancer population. So if we think about the response of the cancer to the local concentration of proliferation signal, it doesn't really make sense that all cancer would have the very same response to a given amount of proliferation signal. So the simplest way in this model to introduce this heterogeneity will be to basically vary these thresholds. So these were the thresholds that would dictate the cell fate based on the local concentration. And you can think of a cell that can sort of downregulate the receptor. So there will be the equivalent of downregulating receptor by moving these thresholds. So you can think of a cell which is what we call a persister cell, which downregulates both receptors, so that no matter under most concentrations of proliferation signal, the cell will be in proliferating mode. Or another strategy could be one where you kind of bring these two thresholds apart so that under most local concentrations, the cell would read these as quiescence, and so it will go quiescent. So we call these persister and sleepers. And so that way we create a full phenotypic space where different cells could basically take different strategies. Obviously, these must come at a cost, as we heard in numerous times. And so this is associated then with the cost of being basically more slowly cycling. But you can be slowly cycling and most of the times in proliferative state, or you can be really fast cycling, but sort of dormant most of the time. And so this creates quite interesting dynamics. And it's quite preliminary results. So I'll just show very briefly a snapshot, and then I'll conclude here. So we just compare control, intermittent treatment, and continuous treatment. And in red will be the ones that we call the persisters. And obviously now the physical location would make a difference. So this needs quite a number of iterations that we don't have at the moment. But just as a proof of principle, we see that spatial segregations of these would definitely make a difference into making intermittent or continuous treatment preferable one to the other. So I think that's the end. So this is the full framework, and obviously the final piece of the puzzle will be to actually include some patient-specific data, as we see that there's this massive variability, and this is really important to calibrate to the individual metastases, especially in the case of a spread metastatic disease over various sites, where different sites have different stromal composition, and you have to come up with one treatment schedule that would overall benefit the entire patient systemically. So I'll just acknowledge, so a lot of the work has been done by my graduate student, Amy, and acknowledging collaborations from past and present, Sandy Anderson at the Moffitt Cancer Center, Philip Maney in Oxford, and Eric Sahai at the Cricks Institute. And thank you very much.